Hi everyone, my name is John Sebastiani, and I'm a PhD student studying marine biology here at the Rosenstiel School. And I want to introduce you to one of my lab mates. This is a Gulf toadfish, and it's the model organism that we use in our lab. These fish are found all over the Gulf of Mexico and the Atlantic coast of Florida and tend to live in shallow coastal waters. They like to hide on the bottom and ambush any prey that comes too close. They'll eat anything that fits in their mouth. And they're unique among fish in that they're one of few species in the world known to communicate vocally. These are very important fish, even though they may not necessarily look like it, because they allow us to study other fish. In our lab, we study their response to different environmental stresses. My research is about how fish respond to hypoxia, or low environmental oxygen, and specifically the role of serotonin in their hypoxia response. Now you may be asking yourself, serotonin, I've heard of that. Isn't that supposed to be a brain molecule? What does that have to do with hypoxia? Well, before I get into that, let me ask you a question. Do you know how long you can hold your breath for? Most people can hold their breath for a minute, maybe two. Let me know how long you can hold your breath in the comments, and we'll see if anyone out there can beat my record of 1 minute 45 seconds. Now, when you start holding your breath, the first 10 to 20 seconds or so, you might feel normal. But then your body starts to change, and you can feel it start to change. Your heart rate will slow, you can feel your pulse in your face, in your extremities. You'll feel lightheaded, maybe some tightness in your chest. What's happening is that you're being deprived of oxygen, and your body needs to make important decisions about how to ration the little oxygen that it has left. The main concern is staying alive. Your body does this by redirecting blood flow to critical organs, like your brain. Your body can adjust the flow of blood by dilating, widening, or constricting, narrowing, your blood vessels. So your body will dilate in some places, like the brain and the heart, and constrict in others, like your digestive system, to prioritize blood flow and make sure that you can survive this hypoxic period. And this is where serotonin comes in. Most people who have heard of serotonin are only familiar with its role in the brain, where it helps control mood, appetite, emotions. Serotonin imbalance in the brain can lead to clinical depression and other mental health disorders, which is probably the most famous role of serotonin. But that's not the only thing that it does. One of its most important functions is actually its role in the blood. Serotonin is a vasoconstrictor, which means that it tightens blood vessels and makes them narrower. Under normal conditions, you have serotonin in your blood helping maintain healthy levels of blood flow, and the serotonin is being held constant. But during hypoxia, when your body is making very specific vasodilations, having so much of this constrictor in your blood might not be such a good idea. And while the mechanisms humans use to control serotonin in the blood are pretty well understood, the methods fish use are not. And this is what I'm trying to address in my research. How are fish controlling the amount of serotonin in their blood? And how does changing blood serotonin keep fish alive during hypoxia? This is a very important area of research because hypoxia is becoming more common in marine environments due to climate change and pollution. We can use toadfish as models for studying marine organisms more generally. Without understanding how fish respond to hypoxia, we risk losing entire ecosystems, including fisheries that humans depend on. Understanding how fish are able to survive hypoxia will allow us to make more effective management plans so that we can better protect them. Learning how fish regulate serotonin will also allow us to better understand how these systems evolved in our own bodies. Findings from this research may be applicable to veterinary or human medicine. Well, I hope you enjoyed learning about my research, and if you're interested in learning more about the Toadfish Lab and what we do, check out our website at www.toadfish.rasmus.miami.edu, where you can meet the members of our lab and read up on our past and current research. Thanks again, stay healthy, and stay safe.